I'm Venkata Mangalampalli, and I'm from Vaccine Production Program Laboratory and Vaccine Research Center, which is a part of NIAID and NIH. And we do the basic research of bringing molecules from discovery research all the way up to phase 1. The challenges we had with this project is something like, if you look at the implementation of the technology that we have used in this study, is like industry uses it at different phases in their process flow. But what we thought is like, what if we implement this in a very early phase, do we have a benefit in reducing our timelines? That was the objective one. The second objective that we wanted to resolve is like, is there a CLONEPIX attribute that we can use in predicting our titers going into the next level? And also like, if this technology can be used for, say, accelerating our cell line development timelines. The methods that we have used, we did not change much with what will come with the methodology of the CLONEPIX. However, what we have changed is the timing where we use the technology. And we see from a conventional standpoint, if it takes almost like more than 7 months to generate a cell line, with our new adapted technology, what we are doing is we can generate clones within 3 months, which is like addition, like we can save time by 3 months. Before we plan, the results are somewhat like mixed up, I should say, because when we started this experiment, we thought that this would fail because of the early implementation and we don't get much of success. However, we could prove that by early adaptation, we can get stable clones which are equally good compared to the conventional method. And we have some clones that are making a productivity of 2 to 3 grams per liter.
Great.
Yes. The other thing that we have seen is like directly comparing the CLONEPIX attributes with that of the titers that are in the 96-well plates, though we did not see much correlation, what we observe is by doing some kind of a normalization based upon the colony size and implementing that normalization and again evaluating the CLONEPIX attributes for that of the titers that we saw, there is a correlation. So in other words, like with this experiment, what we have solved is not only identified a parameter that will be useful in normalizing the titers in the downstream, as well as like a CLONEPIX attribute that you can use to predict which are the high producing clones going forward. Why did we use PDP? Because this is the forum where we see like people from various walks of life. And what we thought was, this technology being there in the industry for a very long time, we thought like folks with different perspectives on analyzing this data would come and then throw their vision on this and then give us a kind of their sense whether the methodology that we applied is the right one, or is there anything else that we have to be looking to compensate for whatever we are seeing. So that was the reason why we chose that this is the best forum to present.